So I'm going to be presenting about severe combined immunodeficiency. It's also known as Scid. But before I get into Scid, I would like to give you a little bit of background on cell communication. So cell communication is the ability of a cell to receive, process, and transmit signals within its environment and within itself. There are two types of cell communication, local and long distance. Local is a signal being sent to adjacent cells within an organelle, and long is a signal being sent from one part of the body to another through the bloodstream. Now, cell communication is also when a message is sent through the body. There is a series of steps that the message goes through to receive a response. This is called the signal transduction pathway. There are 3 steps to this pathway. The first would be reception, where a ligand or a molecule transmitting a signal attaches to a receptor protein, or known as a target cell, on the inside or surface of the cell. The second step would be transduction, where a signal converts to a cellular response in which the binding of the cellular molecule to the receptor occurs. The third and final step is response, where a signal activates the desired response. The cell will now respond in a number of ways depending on the cellular response sent to it. Now, severe combined immunodeficiency, or otherwise known as Scid, is an example of cell communication due to the bone marrow in infants not producing white blood cells, causing the body not to be able to fight off infections. Now, the beginning of Scid it was first reported by Gaussman and Rinkler in 1950. It began with Swiss infants with Scid who were profoundly lymphopenic and died of infections before their first birthdays. With years of differences throughout the patients, a pattern of inheritance was found for Scid patients. Scid is a very rare genetic disease that can cause life-threatening problems within an infant's immune system. It is the largest type of primary immune deficiency, which is simply just a person with an immune system that does not work properly. 1 in every 58,000 babies are born with Scid each year. If the affected infant is diagnosed within the first 3 to 5 months of their life, the survival rate is around 91. However, if it exceeds 3.5 months, the rate drops drastically to 76%. Now, infants appear healthy at birth, but are highly susceptible to severe infections. So they can catch any and everything that is in the household or around them in their settings. Now, more than 80% of infants diagnosed do not have a family history, and gene defects are unknown in approximately 15% of screened newborns with Scid. Now, before I get further into Scid, I would like to contrast with a normal immune system to kind of give you an idea of how a Scid immune system works. A normal immune system is the body's defense against infections. It attacks germs and helps the body to keep healthy. There are parts of the immune system that would include leukocytes, phagocytes, neutrophil, which is a type of phagocyte, a lymphocyte, and then our main focus today will be the 2 types of lymphocytes, B lymphocytes and T lymphocytes. Now, when the body senses antigens, which is any type of sickness or germ that travels into your body, the immune system works together to eliminate them from the body. The B lymphocytes are triggered to make antibodies. These antibodies are also called proteins. They lock onto specific antigens. These antibodies usually stay in the body in order to fight the same antigen again. Some antigens will not enter the body again once they have been fought off, for example, chickenpox, or, as they're saying, COVID. However, although the B cells make the antibodies and can recognize an antigen and lock onto it, they are not able to destroy it. T lymphocytes destroy the antigens tagged by antibodies produced by the B lymphocytes. They will also destroy T cells and are infected or somehow changed. Some T cells are known as natural killer cells, but T cells also send signals to other cells, like the phagocytes, to do their job, which will simply be to eat the germs out of the immune system. Now, in a Scid immune system, the developing babies, their immune systems begin in the bone marrow, as everybody's does, and stem cells then become any of 3 types of blood cells: erythrocytes, leukocytes, or thrombocytes, which, in layman's terms, are just red blood cells, white blood cells, and platelets. However, in Scid babies, both types of leukocytes are affected. The Scid's body either has too few leukocytes or lymphocytes that do not work properly. Due to the immune system not being able to perform correctly, the infections are difficult or even impossible to fight off in an infant with SCID. Now, due to the problem found on an IL-2 RG gene on the X chromosome, critical instructions for making the protein or the common gamma chain is affected. So the IL-2R gene or the common gamma chain is the receptor protein for several different components of the immune system. Due to the defect or absence of the common gamma chain, the T and B cells are not able to get signals to each other to allow the immune system to make antibodies or fight off the infections. This also interrupts the maturation of the two types of cells. So as you can see, this would be a normal immune system where you have an alpha beta chain, which would be these two, and then your common gamma chain in a normal immune system. And it goes through the JAK3, which provides the instructions for the protein. And then it goes down into PY, which is a phosphorylation group, which adds a phosphorylation group to an already existing molecule. And then it goes through the STAT, which kind of gets everything rolling. It kind of sends like a start protein and makes everything start to happen. So normal amounts of T, B, and natural killer cells are made. But in SCID patients, as you can see in this one, the common gamma chain is not there. So there is a decrease in natural killer cells, and there's also a decrease in T cells. So in SCID patients, they are typically diagnosed by the usage of a newborn screening test. So these would be screening tests like for sickle cell anemia and for cystic fibrosis. But babies that do present with SCID through a scene will be referred to a doctor that specializes in immune deficiencies. These doctors will order other blood tests and possibly genetic screening. However, if there is a family history, it is possible for your newborn to be screened before birth. And babies with SCID appear healthy at birth, as I said before, but problems begin soon after birth. And this would be failure to thrive or grow, and chronic diarrhea, frequent, often serious respiratory infections, oral thrush, bacterial infections, viral infections, or fungal infections that are very hard to treat, such as chronic ear infections, sinus infections, skin rashes, meningitis, and pneumonia. Now, the most efficient treatment that has been found today would be a bone marrow transplant. However, if the baby is not diagnosed within the first 3 to 5 months and given a bone marrow transplant, the success rate is very low and could possibly not be effective. There is one other way to treat SCID, which would be gene therapy. However, there have been promising results, but it hasn't been tested enough to be used like locally and more broad. So it would be, there's still research going on about it, so it's not like they would recommend that before they would recommend a bone marrow transplant unless you can't find a match for a bone marrow transplant. And there are precautions that you have to take with a SCID baby because they are so highly susceptible. So children with SCID are placed in isolation to help the prevention of spread of infections, and they should not get live vaccinations as well as anybody that takes care of them or lives in the household, as it can. And SCID patients that do require blood transfusions have to receive specially tested blood that has been tested for antigens and any type of infection within the blood. They also may be stopped from breastfeeding until the mother's milk has been tested to make sure that there is not viral or bacterial or fungal infection, so it will not possibly kill the baby. And there is a movie made about this. There is a couple, but there is one that is most known about it, and it's called The Boy in a Bubble. And this is David Vedder, and he did live his life in a bubble because they could not find a match for his bone marrow transplant. So he became one of the most famous babies with SCID. So they did make a movie about him, and there is another picture of him in his isolation chamber. And then I can't exactly get my hands on an isolation chamber, so I do have a demonstration with a Barbie doll and a hamster ball to kind of demonstrate that for you, to kind of give an idea of how they do have to live their life in isolation. And then this is another baby that I found. I love you. There we go. She does have a brother, and I would assume that it is very hard for them being that young and having to live their life in an isolation chamber, knowing that their brother is on the other side. And that concludes my presentation.